Namaste. Myself, Dr. Reha Rakholia. Today we'll be discussing about two-cell to gonadotropin theory. Two-cell to gonadotropin theory. This is I have taken from ninth edition of Spiroff. Okay, Spiroff. Spiroffs. Okay, two-cell to gonadotropin. What are the two cells? First cell is theca cell. Okay, another cell is granulosa cell. What are two gonadotropins? First is LH, which is acting on theca cell, and FSH. Second is FSH, which is acting on granulosa cell. Okay. So basically, theca cell do have P450 C17 enzyme, which is okay, which is necessary for androgen production from the cholesterol. And it what is not present in theca cell is P450 arrow. So it cannot convert androgen to estrogen. This is not present in theca cell. Now we'll talk about granulosa cell. What is present in granulosa cell is P450 arrow. So from androgen, we'll get estrogen. Okay. What is not present in granulosa cell is 450 P450 C17 enzyme. This is not present in granulosa cell. So basically, granulosa cell is dependent on theca cell for androgen so that it can produce estrogen. Okay. Okay. What is granulosa cell? What is theca cell? This is oocyte. This is cumulus. This is this is mural granulosa. Okay, this is mural granulosa. And outside this is theca cell. Outside the mural granulosa cell is theca cell. This red is theca cell. Okay. Now this enzyme P450 C17 is only present in the theca cell and absent in the granulosa cell. Okay. And this P450 aromatase is only present in the granulosa cell and absent in the theca cell. Now steroidogenesis pathway. See, cholesterol enters into the cell. Like this is LDL cholesterol. Suppose it is LDL cholesterol, okay? Receptor, LDL receptor. If this is LDL cholesterol, this is LDL receptor. Receptor. Now this LDL receptor is given positive feedback from LH. Suppose this is the cell. This is the cell. This is LH receptor. This LH gave this is LH. Okay, this is LH. This is LH receptor. This gave positive feedback to LDL. So LDL cholesterol by endocytosis or internalization. Internalization enters into inside I'm talking about theca cell right now. Okay, I'm talking about theca cell right now. This LDL cholesterol, when got stimulated by LH signaling, then it ends up in endocytosis or internalization, and cholesterol enters into the cell. Now, this cholesterol which is entered into the cell enters the mitochondria of the cell. Mitochondria of the cell. It enters from the outer membrane of the mitochondria to the inner membrane of the mitochondria with the help of star enzyme. In the mitochondria, it is converted to pregnenolone with the help of with the help of 450 SCC. What is this 450 SCC? This 450 SCC is cholesterol side chain cleavage enzyme. SCC stands for side chain cleavage enzyme. Okay. Now, this pregnenolone comes out of mitochondria. Now, all the activity occurs in endoplasmic reticulum. Okay. So, what happens? I'll just Now I'll talk about what happens in the theca cell, endoplasmic reticulum. This pregno, preg, pregnenolone, I'm having cold, that's why. Pregnenolone is changed into 17-hydroxypregnenolone with the help of an enzyme called as 70 alpha hydroxylase. Okay. Then this 17-hydroxypregnenolone pregnenolone is converted into DIA with the help of 1720 lyase. Okay. Then this pregnenolone is converted into progesterone with the help of 3-beta HSD. I have remembered this 3-beta HSD with the help of Hasde, Hasde. Okay. Then this progesterone is converted into 17 hydroxyprogesterone with the help of same enzyme as above, 17 alpha hydroxylase. And this 17 alpha hydroxyprogesterone is converted into androstenedione with the help of 1720 lyase. Then this androstenedione, then this DI is converted to ASD with the help of again 3-beta HSD. Okay, and then this androstenedione is converted into the testosterone with the help of 17-beta HSD, hydroxysteroid dehydrogenase. Okay, and this testosterone is converted to the dihydro testosterone with the help of 5-alpha reductase. So basically, so basically, uh, the we basically two enzyme we have to remember which are very important is 17 alpha hydroxylase and 1720 lyase. Other enzyme is also important, that is 3-beta HSD and 17-beta HSD. Now, what is P450C17, which is also expressed as CYP17, which is present only in the theca cell. I am right now talking about theca cell only. Huh? These contain two main enzymes, 70 P450C17, CYP17 contain two enzymes, that is 17 alpha hydroxylase and 1720 lyase. Lyase means breaking bond. These two enzymes, which we studied right now, 17 alpha hydroxylase, which was converting pregnenolone into 17 hydroxy pregnenolone and progesterone into 17 hydroxy progesterone, because this enzyme do 17 alpha hydroxylation of both pregnenolone, and pregnenolone and progesterone. And these 1720 lyase breaks bond. This is called Caesar reaction also. So, and changed 17 hydroxyprogesterone into DIA and 17 hydroxy pregnenolone into DIA and 17 hydroxyprogesterone into androstenedione. So these two enzymes, the part of P450C17 enzyme, which is only present in the theca cell, not present in the granulosa cell. It is also called as CYP. We take CYP, no? CYP. So CYP17 or P450C17. Now, what happens is, now we'll talk about granulosa cell. In granulosa cell, these two, testosterone and androstenedione, with the help of aromatase enzyme, that is CYP19A1, is converted into testosterone is converted into estradiol and androstenedione is converted into estrone. Okay. Now I have made a picture mnemonic. See, this pregnant woman is alone, lonely. So lone. And she is holding Dia. Dia. That means pregnant, no, lone. Pregnant woman was lone. Now ultimately makes what? Dia. Okay, will you remember? Now another thing is 17 air alpha wolf. Alpha wolf, because we see 17 air alpha wolf is drinking water, means hydroxy, and having lase, means hydroxylase. 17 alpha hydroxylase. Okay. Now this broke, I'll remember by this. 1720 broken. Lyase means broken. Okay. And this one, crying. Crying means rone. Okay. In Hindi, rona. So crying, this rona means progesterone. Progesterone. Okay. Progesterone, because she has autosomal disease. Why is she crying? Rona is there, Rona? Because she has autosomal autosomal disease, ASD, androstenedione. That's why she was crying. So basically, progesterone makes androstenedione, and pregnant woman was lonely, no? So pregnenolone makes dihydroepiandrosterone. Okay, DIA. So like this, I make a picture mnemonic. This lonely pregnant woman I have made. This 17 alpha drinking water and having lase, hydroxylase. Then this change into 17 hydroxypregnanol, and then 1720 lyase. Then ultimately it converted into diya. Diya means lamp in Hindi. Okay, diya. And then this progesterone, Ron means crying. This progesterone, then again 17 alpha hydroxylase and 1720 lyase is converted into androstenedione, means autosomal. I've remembered this by this mnemonic. Autosomal disease is this is having autosomal disease. That's why she's crying. Crying means Rona, and ASD means androstenedione. Okay. Now, as the follicle develop, see this as this we can see by star engine, this cholesterol is taken inside the from the outer mitochondrial membrane to the inner mitochondrial membrane. There, this in the inner mitochondrial membrane, cholesterol is converted into pregnenolone. Pregnenolone. Lone is here. So, what will this make? Lone. If she's lonely pregnant woman, then she'll hold a diya. Okay. Pregnant woman with the help of 450 SCC, side chain cleavage enzyme. 450 SCC. Okay. As the follicle develop, theca cell begin to express gene for LH receptor P450 SCC and beta HSD, hydroxysteroid dehydrogenase. Beta HSD, what beta HSD was doing? Changing pregnenolone into progesterone and diaz into androstenedione. Now, this diagram shows us that LDL cholesterol from the cell of from theca cells is taken by internalization with the help of LH signaling. See, LH receptor is signaling and LDL is taken by internalization. Okay. And like that it is shown. And like LDL is taken by internalization, LDL, but HDL, when there will be ovulation and and luteinization, then HDL will be used to make progesterone in the granulosa cell granulosa cell, but it is not taken through internalization. It is not like LDL. It is not taken through internalization, endocytosis. It is not taken by endocytosis. HDL is not taken by endocytosis. When after ovulation, luteinization occur of granulosa cell, then HDL is not taken. HDL is taken for making progesterone, but not through internalization. Okay. After ovulation, this is the line of Spiroff. Luteinized granulosa cell can use HDL cholesterol. Okay. But unlike LDL, HDL is not internalized. Instead, cholesterol ester from HDL are extracted at the cell surface and transferred into the cell. See, this diagram is showing the same. This diagram don't have anything to do with two cell to gonadotropin theory, but see, it's showing that progesterone synthesis occur after ovulation. Okay, luteinization of the cells of occurs after ovulation. So, these three HDL, LDL, and cholesterol esters contribute in making progesterone. See, HDL, LDL is stored cholesterol ester is contributing in making progesterone. Same, it is showing luteinized granulosa cell is making estradiol and progesterone from the cholesterol. Now, two cell, two roles. I have already discussed this. The granulosa cell has FSH receptor. Granulosa cell has FSH receptor, and theca cell has LH receptor. Okay. So theca cell, this theca cell convert androgen cholesterol into androgen, and this granulosa cell convert androgen to estrogen. In this, P450 aromatase enzyme is used to convert androgen into estrogen. In the theca cell, P450 C17, which contain two enzymes, 17 alpha hydroxylase and 1720 lyase. And another enzyme is P450 SCC, which is involved in changing cholesterol into pregnenolone. Okay. Now receptors already discussed. FSH receptor are present in granulosa cell. LH receptor are present in theca cell, and approx 20,000 LH receptor present per cell. Okay. Function. Granulosa cell I've already discussed. Granulosa cell forms estrogen from androgen with the help of P450 means aromatase enzyme, and theca cell produce androgen with LDL cholesterol with the help of two enzymes, P450 C17, P450 C17, which contain two enzymes. What are those enzymes? 17 alpha hydroxylase and 1720 lyase. Let me say, these androgen are precursor for estrogen synthesis. Now, these androgen will move to granulosa cell and act as a precursor for estrogen synthesis. Aromatase activity is high in granulosa cell as it has to make estrogen, but minimal in theca cell. Okay. This expresses, I have this everybody knows. This expresses P450 this expresses granulosa cell expresses P4 aromatase enzyme, and it does not expresses P450C17. Okay, does not express P. And as the follicle mature, as the follicle mature, granulosa cell will have more expression of P450 arome, and uh, theca cell will have more expression of P450C17. So basically, these two enzymes are signs of maturity. This enzyme is signs of maturity for granulosa cell, and this enzyme is sign of maturity for theca cell. Okay. Now we are talking about granulosa cell, small and large follicle. If granulosa cell is small, then this small follicle convert androgen to dihydrotestosterone, 5-alpha reductase form. Okay, 5-alpha reductase form. If the follicle is small, but if the follicle is large, it will produce estrogen. This I have already discussed, that theca cell expresses LH receptor P450 SCC 3 beta hydroxy dehydrogenase as the follicle grow. Microenvironment. What happen is A granulosa cell in large follicle changes androgen environment, dominant environment into estrogen dominant environment. Okay. So this estrogen dominant environment is essential for growth and development of follicle. Okay. And it is dependent on growing sensitivity to FSH, growing dependent on growing sensitivity to FSH, which is brought about by FSH itself and estrogen. So basically, large cell has, granulosa cell has estrogenic microenvironment. Microenvironment. Okay. Now it is talking about small antral cell and large antral cell. Okay, small antral cell in granulosa theca interaction is not fully functional, while in large antral cell it is functional. So there is accelerated estrogen production. And in androgen conversion, small antral cell convert and androgen into potent 5-alpha reduced form, means dihydrotestosterone, while here in the large cell, it is converted into estrogen. Microenvironment in small cell, it is androgen dominant. In large cell, it is estrogen dominant. I've already told you how it is estrogen dominant, because the growing cell will have more FSH sensitivity because of FSH itself and estrogen. I'm having cold. Huh? FSH sensitivity. A small antral